Alrighty, hi everyone, I'm Sarah. Are we having a great night tonight? Yeah, I know I am. This is actually the most human contact that I've had all week. And yes, I wish I was joking. So I'm a PhD student at the University of Queensland, which technically means that I'm training to be a doctor. Well, not the kind my parents were hoping for, or the kind that is useful on a plane. But hey, at least I get to eat unlimited smashed avo because I'm not buying a house anytime soon. In fact, I was actually thinking about wearing this for Halloween just to haunt my parents that little bit more. But in addition to being a stay-at-home daughter, I'm also a biomedical scientist, which means that I get to do a bunch of serious lab stuff to try and improve human health. And if you're from work health and safety, no, you didn't see these pictures. But more specifically, my PhD project is on ovarian cancer and how it interacts with the immune system. Now, when people hear that I'm a cancer researcher, I often get the same 3 questions. Do you find it hard? Are you like, smart? And the dreaded, so when are you gonna cure cancer? To which I say, Stacey, babes, if I were close to curing cancer, do you think I'd be here, trying my hand at stand-up comedy? But don't worry, if this doesn't work out, I think my parents' dog is looking for a stay-at-home sister. And I promise she loves me, it's just the angle, trust me. So I do something called basic research. And I know what you're thinking. Basic sounds like I wear Ugg boots and analyse star signs. I am a Cancer Leo cusp, by the way. But in my field, basic means that we're trying to answer the fundamental questions. And we're trying to, I guess, piece together this puzzle of how cancer behaves, how it resists treatment, and what genes and pathways are involved. And so this kind of work is actually really important for ovarian cancer. And if I leave you with one message tonight, I want it to be that we need to invest more research into this disease, because ovarian cancer is incredibly deadly, and compared to other cancer types, it's hard to treat, and survival rates haven't changed in over 30 years. And so I'm actually really grateful for this opportunity to tell you a bit more about the research that we're doing to try and uncover why ovarian cancer resists treatment and what we're doing to try and flip that narrative. But first, I need to bring you back to, sorry, I forgot about that slide, bring you back to your immune system. So immune system's actually pretty cool. You have these cells called NK cells and T cells that have this natural ability to be able to detect and fight cancer. So they're basically your body's Avengers, except with fewer hot guys named Chris. But the thing is that cancer fights back. And it creates this super toxic work environment. I mean, you guys know the type. Unironically uses Teams. Pretends carrots are a fun snack. Or even worse, is a carrot on a power trip. But I digress. The point being is that cancer creates a really unaccommodating environment for your immune system. And so we can describe this, I guess, dynamic between tumour cells and immune cells through this concept of hot and cold tumours. Yeah, like that Katy Perry song from MasterChef. So as I mentioned, we have hot tumours, which are full of immune cells. I mean, think about the Valley on a Saturday night. It's messy, it's chaotic, you've got punch-ups between T cells and tumour cells. I don't know, guys, use your imagination. Like I said, I haven't been outside in a while. But in contrast, we have cold tumours. So these guys are like immune deserts. So you could even think about Toowoomba, also on a Saturday night. But why does this matter? Because we have these really exciting new therapies called immune therapy. But the thing is that they only work if the immune system is already present. So take melanoma, which is an example of a hot tumour. Immune therapy has completely changed the game. So today, over half of patients with advanced melanoma live 10 years or longer, when just 15 years ago, that number was 6 to 9 months. But in contrast, that we just don't see the same thing with ovarian cancer, and it's because it's typically a cold tumour. And so these potentially game-changing treatments just don't work. So that leads me to our lab's big research question, which is how can we heat up a cold tumour, and can we use that to try and improve immune therapy? So to do this, I first turned to something called Biomedical, which is basically like this weird love child between biology and coding. And I initially thought it would be something like this. But in reality, it turned out to be something more like this. And so I guess, how did I get here? Well, what I had to do was analyse data sets from hundreds of ovarian cancer patients and look for patterns. And specifically, I was looking for genes that were associated with these cold tumours. Now, the problem is that there's about 25,000 genes in the human genome. So to be able to narrow this down, I basically did a scientific version of Tinder. I set my criteria and I got swiping and found a few eligible bachelors. What's interesting is that none of these genes had been studied in the context of ovarian cancer immunity before, and on top of that, they were associated with cold tumours. And so in my lady lizard brain, this translated to sketchy, mysterious, and full of red flags. Just my type. And so, of course, I was hooked. And so I wanted to figure out what these genes are actually doing. So to do that, I took ovarian cancer cells, I then blocked the genes one by one, and then ran a super expensive experiment called RNA sequencing, which basically gives you just this alphabet soup. And it was really only through what I can describe as statistical witchcraft that this turned into something meaningful. So what did I actually find? And yes, I know, I bought a graph, but in my defense, it is a bar graph, and bar graphs are pretty hot. And no, Stacey, I'm not open for feedback. And so what I really want to bring your attention to is that when I blocked most of these genes, we actually saw that the tumour was predicted to become more hot and respond better to immune therapy, which is a really exciting finding. But it wasn't always a win, so yeah, that little blue bar that you see there, when I blocked that gene, it actually made the tumour colder. And so yes, there was that one time that I made cancer worse. And if you want to know more, you can ask my therapist. But that's science. Sometimes things work, sometimes they don't work out. But we did find a few solid leads, which brings me to Chad. No, not that Chad. This Chad is a transcription factor, although the resemblance is kind of uncanny. As a transcription factor, Chad doesn't actually do anything himself. He just tells the other genes what to do and somehow makes everything worse. Like I said, classic Chad. But at this point, all of my data was very statistical, probabilistic, and I wanted to figure out whether Chad actually does something inside a real tumour. So to test this, what I did was I basically got ovarian cancer cells, blocked Chad, and then transplanted them into mice. And the results were honestly pretty remarkable. We found that when we blocked Chad, the tumours were not only smaller, they grew slower, but those Avengers that I was talking about, the T cells, the NK cells, suddenly started showing up, and the environment started to resemble a hot tumour. And this is actually a really exciting finding because it opens up new therapeutic opportunities. Because if we can figure out how to block Chad in patients, then maybe we'll be able to improve immune therapy. But to be able to do this, we need to figure out how to manipulate Chad in patients. So that raises an important question. Can we drug Chad? Which, now that I've actually said that out loud, sounds a lot more illegal than I thought. So if you're Chad and in the audience, I'm sorry. Please don't send me a restraining order. I've already got too many of those. But this is what my PhD is now focusing on. I'm trying to figure out what makes Chad tick. Who are his enablers? You know, who's his Brad, his Thad, his Jaden, his Brayden? Can we target those instead to indirectly get to Chad? Hopefully I'll be able to get back to you in a few more years. If I'm looking a bit frazzled tonight, it's because Chad's actually turned out to be a bit more of a slippery seal than I initially thought. But that's the thing, science is slow, it doesn't work out sometimes, but at the same time, it's really hopeful. Because if we can figure out what, I guess, genes and regulators are contributing to these cold tumours, then we can give immune therapy a real shot in ovarian cancer. And I feel incredibly grateful to be able to contribute even just one piece to this puzzle. And so, no, Stacey, I haven't cured cancer yet, but I think I have found a Halloween costume that's scarier than my housing prospects. Thank you, everyone, and I hope you have a great night.